education about Friedreich's ataxia, what's going on in research, clinical trials, and we also want to give you the opportunity to meet the community and build relationships and learn from one another. And so your name tags have colored lanyards on purpose. So the folks who have green lanyards are people who have recently been diagnosed with FA or are new to our community. And the folks who have blue lanyards are folks who are not new to our community. So everybody with a blue lanyard needs to meet somebody with a green lanyard today. That is a requirement. Red lanyards are for FARA staff. So if you have any questions throughout the day, there's several of us here. We're wearing red lanyards, so please find us. And then the folks who have black lanyards are our speakers, folks from industry, academic, researchers. So do not be afraid to talk to them and ask them your questions. So those are sort of our overarching goals for today: education, networking, learning from each other. And so to start us off this morning, I'm really privileged to work with Dr. Perlman for many years. She is one of the principal investigators in our collaborative clinical research network. She participates in our natural history study, all of our clinical trials. She is really the FA expert here on the West Coast and nationally and internationally. And we are really privileged to work with her and her team and have them help us host this symposium every other year. So Dr. Perlman, thank you.
I have half an hour to tell you everything there is to know about Friedreich's ataxia. I think we'll focus on the key points that you always have to keep in mind. So whenever you're in a situation, meeting someone, bumping into somebody from a drug company, you'll have the questions that you need to ask at the tip of your tongue. And I think that's important now, as I was discussing with some people before, that 10, 15 years ago, there was one drug trial every 5 years. It was growing, but very, very slow. And I think we'll see today that the pace has definitely picked up. You know, research and development, finding ways to intervene in the processes that result in the clinical condition of Friedreich's ataxia are exploding. And you know, we need to have a voice in that so that we can keep them moving forward efficiently. You know, sometimes you hear about a new drug that's in development, and 2 years later it's still in development, and a year after that it's still preclinical on the pipeline on the FARA website. So perhaps we can also give, you know, the bigwigs at the companies and FARA itself ideas about how to speed up trials to get the information we need quickly so we can carry the drug forward or just put it on the shelf or drop it in the trash, so that we don't have to keep looking at it on the pipeline for 5 years and wonder what it's doing. So I have to have my disclosures. In the last 5 years, I decided to reinvent myself as a clinical trialist. So I'm now running clinical trials in Friedreich's, in Huntington's disease, in spinocerebellar ataxia, ataxia-telangiectasia. Yeah, there's a lot of overlap. You know, you learn from one trial how to make another trial better. I have a wonderful team working with me. Why don't you guys all stand up over there? Anybody who's geographically near us and wants to find out more about what we're doing, you know, you can interact with them, and potentially we can enroll you in something that will benefit you, but benefit the community as well. So there's been an avalanche of information. Key points that you see in a lot of press releases. 2 in 100,000 people have Friedreich's, as many as 20,000 worldwide, a very rare disease. It's even rarer in Asia and sub-Saharan Africa. If you Google Friedreich's ataxia, there's 500,000 hits of all types of input, science and popular. 3,500 scientific articles in the past 125 years when Friedreich's ataxia was first described. 2,200 articles in the 25 years since the gene was described. 1,200 publications in the last 10 years alone. So there is increasing scientific interest. And science is not going to move forward without our participation. There have been close to 40 drugs in the FARA pipeline over this period of time, but there are currently no approved treatments for free drugs. Everything that we learn, every bit of knowledge that we as a group can bring to FARA, can bring to the drug companies, is going to open up new avenues for research. And I'm going to demonstrate that. I put that in red because in each of the slides I'm going to show now, I've highlighted in red an item of information that has powered up clinical research. So, upcoming agenda, we're going to be hearing about molecular mechanisms and avenues of therapeutic intervention. That is, how do you develop a drug that's going to target the right thing and have the right effect? We're going to hear from people who are conducting clinical trials from pharma. We're going to look at a couple of very exciting interventions. How to make the mutant gene for taxing protein behave like a normal gene. There's a number of drugs in the pipeline that could do that. And then also the final scientific group is going to talk about gene therapy, gene replacement therapy predominantly. So we can replace the gene, that's obviously everybody's holy grail. We can make the mutant gene behave more normally. We can make the frataxin protein that is being produced have higher levels, hang around longer, work better. And all of these wonderful basic science ideas have come through the clinical research that we have all participated in. So, overview. It's been known as a unique neurologic disorder of children, since the 1860s when it was first described. It has very specific neurologic and neuropathologic features that were described by Niklaus Friedreich, so that an examination of a young person with a balance disorder, back in the late 1800s, they could say this is Friedreich's ataxia. It looked very, very specifically. I have a little picture there, which is a cross-section of the spinal cord showing some of its inflow pathways and pathways that go up to the cerebellum. The ones that are targeted by Friedreich's, the cerebellum itself to some extent, the dorsal root ganglion, which is that little knob right there, brings signals in from the periphery and sends them up to the cerebellum. And then the descending pathways in the spinal cord that bring the signals back to our muscles for performance. So each of those tracts that I have listed there underlies a specific symptom or sign that we can find in people with Friedreich's. The incoordination, the change in sensitivity, the absent reflexes, the optomentos. The gene was discovered in 1996. It was reported by Dr. Pandolfo's group. Genetic testing that resulted from the discovery of the gene has made it possible to clinically diagnose Friedreich's ataxia. You have a gene test, it's positive, this is what you have. It also revealed that adults could develop Friedreich's ataxia, that it wasn't just a disease of childhood. It revealed that you could have Friedreich's ataxia and still have reflexes with the reflex hammer. Previous to this, if you were over 25 and had reflexes, by definition you did not have Friedreich's. But with the advent of the gene test, we were able to recapture all of those individuals. But we also found that some people that we had diagnosed clinically as Friedreich's, they looked just like Friedreich's, had a negative gene test. So it was about 10% of people looked like they had Friedreich's but did not genetically. And another 10% didn't look like Friedreich's, but indeed that's what they had. So we've been able to enlarge the group. We've also been able to be more specific and weed out, as it were, people that still needed to do more work in finding their specific diagnosis. The gene mutation, GAA expansion, so there's probably some science that we're going to hear from some of the presenters later on. What it is a segment of the DNA in the gene for frataxin protein is expanded, is enlarged. It blocks the ability of the gene to make messenger RNA, which makes the frataxin protein. So there's a roadblock, and the little picture here shows that a normal gene is going to make so many units of frataxin protein, while a mutant gene may make one or none. So there's an overall decrease in the amount of frataxin protein that is available. Carriers have one mutant gene. Two carriers marry each other, and that's the top picture there. They're purple because they have a normal gene which is blue, and they have an abnormal gene which is red, and you combine them and it's purple. If they have 4 children on average, or 400 children, or 4,000 children, 25% are going to inherit both bad genes, 25% are going to inherit both good genes, and 50% of their children on average will be carriers. They'll have one bad gene. And early studies done of carriers, is there any risk in being a carrier? We know that for certain diseases, if you're a carrier, you're at risk for whatever. If you have 2 bad genes, you have the disease itself. They were never able to fully document that carriers had any increased risk for neurologic issues or orthopedic issues or heart issues or diabetes. But what we did learn, and that's in red, that you only need the activity of one normal gene to be normal in all of the areas that are stressed when you have 2 bad genes. So if we're thinking about boosting frataxin protein levels by any of the mechanisms we're going to be hearing about, they don't have to come up to 100%. They could come up to 50% or 45% and essentially restore the system to normal. So frataxin protein is required in mitochondria. And I have a little picture here that 2 little pictures I took from this online article that I found. Don't you love the internet? So these were illustrations from this particular publication from 7 years ago. And this is a picture of a mitochondrion. It's got 2 membranes that are kind of envelopes, and the inner membrane, which has these kind of folds in it, is where energy production occurs. You need frataxin protein for the mitochondrion to make energy, and the mitochondrion, is here. This is a nerve cell, and it sits in the cell body of the nerve cell. It's also down here at the ends, where the nerve cell is communicating with other nerve cells. Makes energy to support all of those processes that the nerve cell, heart muscle cells, pancreas, and basically all the cells in our body require to a greater or lesser extent. The brain has, it does not have the ability to store energy. So if there's even a little decrease in energy production, the brain will feel that very quickly. Similarly with the heart muscle is constantly working, and Dr. Kane is here. He can correct me if I'm oversimplifying. But the heart muscle needs energy, and it needs it reliably. Heart and blood muscles, they can store energy. They can operate without oxygen on anaerobic energy production for a while. And that's why you get muscle cramps. But the brain and the heart really require constant supply of energy, and the pancreas also for reasons I don't understand. Dr. Kane, being an internist, will understand it better than I. So the frataxin protein produces iron sulfur clusters. It combines iron and sulfur, which then go on to make the mitochondrion able to produce energy. It's called a cofactor. So no frataxin protein or lowered levels, lowered amounts of iron sulfur clusters, lowered energy production, free iron floating around, which is very toxic and can create free radicals, can damage the mitochondrion, which further decreases the ability of nerve cells and heart muscle cells to function. Testing that may be used in the diagnosis of Friedreich's has also been explored by several of our clinical research sites as possible clinical biomarkers. How do you tell if a treatment that you're developing is actually helping? If it's for symptoms, well, your symptoms will be better. Think if you have a tremor, you can take tremor medicine and the tremor will be reduced usually fairly quickly. If you're having pain, you can take medication for pain or muscle spasm and it will work fairly quickly. But if you're looking at drugs or treatments that can slow the progression of a disease, how long do you have to monitor somebody on that research treatment to tell that it's really working? So we do use biomarkers in clinical trials, and anybody who's participated in clinical trials knows the variety of things that are used. Many of the biomarkers are tests that are used to make a diagnosis. The EMG, measuring electrical muscle activity. Nerve conduction studies, how quickly the nerves can send a signal. All the heart studies that we do for clinical management can also serve as early markers of a treatment that might be helping the heart. We might see benefits within a month or two as opposed to 6 months or a year. There's always blood work, and there's more and more blood tests being developed to try to find the ideal biomarkers. X-rays for the scoliosis, I think, most Friedreich's patients have had at least one round of those. Brain imaging, can be helpful in ruling out other things that can cause ataxia in a young person. But there are very refined brain imaging studies, PET scans, MR spectroscopy, MRI scans, and CT scans that are being used as biomarkers. Can we show improved activity in the cerebellum on one of these brain imaging studies that might appear within 6 weeks of starting something, as opposed to waiting 6 months or a year? Genetic testing for the GAA expansion is the standard genetic test that is used. 5% of people will have one expanded segment, that little enlarged area, and what's called a point mutation, or a small tweak in the coding in the other gene that may totally disable the other gene, may disable it a little bit. And there's a wonderful article that was published under Dr. Lynch's guidance, also I think with our group in Australia, looking at the different small tweaks and how damaging are they? How damaging are they when they're paired with an expansion on the other gene? How do they compare to somebody who has 2 expansions? So we are still learning a lot about how to use these testing to improve clinical care, but also to improve our ability to do research efficiently and quickly. So there can still be a delay in diagnosis if you present as an older person. I think the oldest person I met with Friedreich's developed symptoms at the age of 69. And I know that Dr. Lynch, I think, has older people as well. Very small mutation, and it took all those years to really build up the changes that became apparent. But once you are diagnosed, these are the things you should do. And I think FARA is going to have some of these slides available. But this is a handout that I've given many of my patients, just so you can check it off. This is what I can do now. I have this diagnosis, this is what I can do now. You can participate in the natural history study. You can give blood samples, you can give cheek swab samples for the research that's going on. Your parents can give blood samples and cheek swab samples. You should see a cardiologist once a year. I know back in, when I first saw my first patient with Friedreich's in the late 1970s, we had a consulting cardiologist, and he said, after the age of 40, don't bother with the cardiac testing. Whatever they're going to develop early. They don't have changes by the time they're 40. It's going to be aging. They're just going to have normal heart things like everybody else. Well, that's not true. And I think Dr. Payne is going to be able to enlarge on that. So, we are recommending that you establish a relationship with a cardiologist, even if your heart is really good, through your 20s. It's important to continue to follow it, to catch any changes early. I think the biggest progress we've made in Friedreich's is the ability to manage the heart problems. In individuals who have symptomatic heart problems, the ability to manage them is much better than it was 30 years ago. Some of the other things we use to manage, physical therapy, rehabilitation strategies, which are definitely on the list. You need to exercise. We knew that 30 years ago. I don't know that we've improved on that much, except there's more fun ways to exercise now than perhaps there were back then. You can consider antioxidant supplements, and that's an excellent point for discussion when we start talking about the drugs that are in development and how they relate to some of the older antioxidants that were the first things that were tried. If you feel a need to try a new supplement, lion's mane, or some new idea, I would like to, have somebody put me on erythropoietin, or, my primary care doctor is willing to try, this unusual thing. There's one case report in the world, and I want to try it. Be sure that your Friedreich's doctor knows what you're doing. I had a little surprise with a patient enrolled in a clinical trial, not Friedreich's, something else, and his screening blood work showed that his blood was thinner than I would have expected. He wasn't taking aspirin, he wasn't on blood thinners, no heart problems that would have entailed any drug that would have thinned his blood. He was taking lion's mane. And I looked it up, and it can thin your blood. I did not know that. So I must admit, every time I interact with a patient and family, I learn something new that I can then pass on to other people. Good resources, I think we all know, the resources, the FARA website, FAPG, Facebook groups for patients with free breaks, ways to network with the community and also share ideas. I am allowed to observe the parents' listserv, where it's kind of an email posting thing. I don't know exactly what it is. I'm not that familiar with social media, but I can look at the discussions that the parents have, and they talk about very important things. Something that maybe I haven't thought about in 2 or 3 months because nobody's mentioned it in my clinic. And now the next person that comes in, I might ask them, well, I heard, I won't tell them where, that this is sometimes a problem, and is this ever been a problem for you? So I think, patients, parents, families, doctors can share information that will benefit all of us. Be sure that you're signed up in the registry so that you can be notified about upcoming clinical trials that you might qualify for. And take care of your general health. Having free breaks does not make you immune to other health problems. Yeah, you may have a new symptom and it's easy to write it off as due to free breaks, but make sure that it's investigated and that you're not missing something that needs to be treated in a different way. My doctor doesn't know about free breaks. I hear this very often. The FARA website, curafa.org, has posted the 200-page manuscript of clinical care guidelines. We're not going to go through each one. No, we're not. But there is a 12-page executive summary which summarizes for each of the symptom areas: ataxia, weakness, neuropathy, spasticity, restless legs, diabetes, pregnancy, quality of life, a little summary about appropriate interventions, questions to ask and ways to deal with it, and then the longer detailed manuscript follows that. I frequently go to that website, will cut and paste out a segment about the heart or about the bladder so that I can share it with the patient and share it back with the patient's primary care physician. And they can go directly to the website as you can and read it in its primary form. So, the CCRN, Collaborative Clinical Research Network, I think that's what the initials stand for, funded by FARA. These are the sites that are actively involved in the natural history study. There's sites for drug trials, there's sites for biomarker studies, but there's still nothing in the Pacific Northwest or Texas. So unless FARA is trying to recruit some sites, we still have some big holes in the map requiring people to travel fair distances. So when you are thinking of participating, even in a once-a-year natural history study or in a drug trial that may be more demanding of your time, the sacrifice that you make to travel 1,000 miles every 3 months, to spend a day in a research setting is invaluable. Without all of our participation and the participation of patients and families, we will never get drugs approved for free breaks ataxia. So I can't thank the patients and families that I work with enough for everything that they've taught me and for their willingness to support this effort. Rare diseases often get overlooked. One person who's a cousin of your brother-in-law, and they have a rare disease, and they're not really sure what it is, and oh, that's so sad. But it's, from our perspective, we can develop drugs that will be disease-modifying, and I think we can even talk about the cure FA. Yes.